Okay, hopefully everyone is looking at this practice warm-up problem on the board right now. I'll give you a couple minutes to take a look at this before we begin. Okay, hopefully that was enough time for you to take a look back in your notes and think about our hormone ADH, antidiuretic hormone, which affects our principal cells in the distal tubule, late distal tubule, and the collecting duct. So this is how we're controlling water balance, and therefore also how we're controlling blood volume, because blood volume is directly related to blood pressure. We're also controlling blood pressure with this hormone ADH. So when we're thinking about ADH, we want to think about what's going on in those cells. So ADH is coming from our posterior pituitary and responding to either, and I'll go back into our previous slide where we had these diagrams, right? We're responding either to high osmolarity, so like lots of ions in fluids reaching our osmoreceptors in the hypothalamus, or we're responding to low blood pressure directly, right? Low MAP, we have baroreceptors, so pressure receptors. We also have volume receptors that would tell us about this. This all tells our posterior pituitary to release ADH. Okay, so that brings us to our cell, right? So as we go into our late distal tubule or in the collecting duct, we have these cells called principal cells. I'm just kind of highlighting right now that they're called principal cells because we haven't seen that word in a while. And that's the name of this specific cell that's responding to our regulating hormones, really. Okay, so ADH is along in our plasma, in our bloodstream. Okay. Someone joining us in the waiting room, sorry y'all. Okay. ADH is coming along in our plasma as binding to a receptor, G protein coupled receptor on our basolateral membrane. We remember that the basolateral membrane, it always has aquaporins, specifically aquaporin 3 here. Okay, so that's also there, but it doesn't always have aquaporins on the apical membrane. So when ADH binds to a receptor on this basolateral membrane, what we're ultimately going to do is tell the cell to take some aquaporin 2 molecules that it's already created and insert them into the apical membrane so that we can take in more water. All right, that's our ultimate goal here. So we do that by activating a second messenger system. All right, we make this molecule cyclic AMP. Then we have a flow kind of enzymes and proteins, specifically protein kinase A, that tells our cell, go insert these aquaporins on the apical membrane. So ADH binds to a receptor, triggers the insertion of aquaporin 2 on the apical membrane. Right. When we do this, once we've inserted aquaporin 2, we now have an aquaporin on the apical membrane and an aquaporin on the basolateral membrane. The aquaporin 3, right, was the one that was already there. It's there the whole time. Okay. But once we have a door, essentially, on either side of the cell, this allows water to flow all the way through. So when we have ADH, when we have aquaporins on both sides of the cell, water is able to move into the cell, and then it's allowed to continue out of the cell on the opposite side and ultimately to go into the plasma. Now, the ultimate result of that, right, when we have water leaving our tubular fluid, leaving what is essentially our urine, right, leaving here, right, so we're going to have less water here, meaning we're going to have lower volume urine, right? So when it's lower volume, there's less water in it. There's less water watering down our waste products here. So it's going to be low volume. It's going to be highly concentrated. Now, all that water is going to be over here, right? So the converse of this, the opposite side of this, if we were to ask about what's happening to the blood pressure and the blood concentrations, right? What we're doing here is we're watering down the plasma. So on the opposite side, we'd be increasing volume in the plasma, right? And then decreasing the concentration, so specifically decreasing osmolarity of the plasma, which means that we're basically going to be turning off the two signals for ADH release. Okay, right? Because our two signals telling us to release ADH were either the blood pressure or the osmolarity in the fluids touching the hypothalamus or touching our baroreceptors. And as we readjust, we turn those off. We're no longer receiving those signals. Okay. Before we move on to our control of sodium and potassium, any questions remaining about this water balance that we did on Wednesday? Because we're kind of going to be building off of this with one of the systems that we're going to see today. Okay, so teaser for maybe 15 minutes from now, right? We're going to see that aldosterone and this renin-angiotensin-aldosterone system is actually going to affect this whole ADH release as well, which is part of why it's important that we cover ADH first, because we're going to talk about something that triggers this in addition to blood pressure and osmolarity. So we're kind of moving backwards down the system in a way. Okay. So for today, we're talking about two hormones, aldosterone and atrial natriuretic peptide, or ANP, that are going to be regulating solute concentrations. So they're really going to be regulating plasma sodium levels, but we'll also see that they will be involved with potassium balance as well. So we're going to take a look at this linkage between sodium levels and water balance, which is why it's important that we understand ADH before we get there, because these two systems are kind of going to work in conjunction to influence mean arterial pressure. Okay. Realistically, we're not going to talk about calcium balance, so you can cross off this last one. I want to make sure we have time to go through everything kind of slowly. So we're probably also going to push acid-base balance, the final the final presentation in this unit. We're going to probably push to the next exam. I don't expect we'll get much into that today, and I want to make sure we have time to review. Okay. So a super important function of the kidneys overall is to regulate sodium and calcium, or sorry, sodium and potassium, and to regulate pH. Like when you are in a clinical setting, that's often what we'll be trying to treat with different medications. So it's really important to understand sodium in particular. Sodium is the really big thing. You've probably encountered people who have, high blood pressure problems and are told to reduce their salt intake, right? Part of that is because this linkage between our kidneys, how they're balancing sodium, and how that's affecting water reabsorption and therefore blood volume and blood pressure. So all these things are kind of linked together. Sodium is the most important thing that we will really be looking at. Not a big shocker, right? We've talked about sodium a lot in this class. So we have words for high levels of sodium versus low levels of sodium. So we're using our normal kind of prefix for high, right? So hypernatremia, right? So kind of nifty here. I don't know if this is on purpose, probably, right? So here's our sodium in the middle, right? So hypernatremia is for our sodium ion. Emia usually refers to the fact that there's something in the blood, right? Like, remember the word anemia, right? Anemia had to do kind of with hemoglobin and iron in the blood. All right, so hypernatremia, it's high sodium in the plasma. Hyponatremia is just the opposite. So hypo meaning low. Here we have our sodium in the middle. And here we have our indication that we're talking about blood. I don't always know if it's helpful, by the way, that I circle these word roots for you, but I do want to tell you why I do this consistently. It's basically so in the future, if you hear a term that you don't recognize, you can kind of guess. There are a lot of weird medical terms using all these Latin and Greek roots. And so it's helpful to kind of be able to guess where you should be looking, even if you don't know the specific word. And that's a large part of why we do what we do in the anatomy lab as well. I know I hear back from students, right? You're not going to remember all the things you memorized, and that's true. But it's important to be able to look things up quickly. So the goal of lots of the things we do is to give you familiarity with terms so that you're better able to look these things up and learn them really quickly in the future when you're pressed for time and don't have a lot of time to be able to really read things in depth. So that's why I circle these things for you, because we are not going through a comprehensive list of all the things that could be wrong with someone, right? Okay, back to sodium. Sodium is our primary solute in our extracellular fluid, right? It's one of the main things we track, and it's really important for our normal osmotic pressure. So we've talked a lot about how proteins affect oncotic pressure being osmotic pressure specifically caused by proteins. But most of the time, a lot of the osmotic pressure is because of sodium. And we've seen in a lot of places, right, how we control the movement of different solutes and the movement of water across cell layers by controlling the movement of sodium with our sodium-potassium pumps. So this shouldn't come as a big surprise, right? And when we talked about neurons in specific, right, and also our muscles, we looked at how action potentials are really dependent upon our concentration of sodium and the movement of sodium into the cells, right? Depolarizing, creating these action potentials that ultimately create the release of our neurotransmitters or actually create muscle contractions, right? So it's really important to have a stable concentration of sodium in our extracellular fluid, because it's the gradient, right? The difference between the inside of the cell and the outside of the cell that creates movement of these ions, right? The movement of sodium is dependent on where you have more versus less sodium. So if we mess too much with the sodium concentration in the extracellular fluid, we're messing with the gradient across cell membranes, which means we're messing with the movement of sodium across cell membranes, which means we're potentially messing with action potentials, therefore messing with neurotransmitter release, therefore messing with the function of our muscles. So it's a lot of steps, but sodium is really important to how your body functions. So it's really important to maintain appropriate sodium concentrations in the plasma and therefore in your interstitial fluid. Okay. So we're controlling the amount of sodium by controlling the amount of sodium reabsorption in the renal tubules. So early on in the renal tubules, so like in the proximal tubules, the way we get sodium in there, right, is it's being filtered at the glomerulus, right? So we remember that at the glomerulus, right, we have a certain GFR, a certain glomerular filtration rate, and everything except our proteins and our red blood cells are basically traveling into the nephron because we have a lot of sort of gaps in those capillaries, right? They're specifically structured to allow free filtration of basically the full contents of the plasma. So this free filtration is happening at the level of the glomerulus, so right at the beginning of our nephron, right? Sorry, that's a glomerulus, right? The capillaries swirling around, going in to Bowman's capsule, specifically Bowman's space, following the filtration pressure, right? Which we can define with our Starling forces, our hydrostatic and oncotic pressure for both the capillaries in the glomerulus and the fluid in Bowman's space, right? Kind of fighting against each other, depending on which ones we're talking about. But we have sodium filtering in roughly the concentration it is in the plasma into the glomerulus. Then we enter our proximal tubule, proximal convoluted tubule, right? So I'll draw it convoluted, right? And we remember our proximal tubules are basically bulk reabsorbers, right? So there's a lot of reabsorption, so absorption back into the bloodstream happening here. So we have a bunch of reabsorption here. We go into our loop of Henle. That's not specifically traced out on here, but I think it's worth doing. Sorry, I'm going to cover some of these words, but you have them on your PowerPoint, so that's okay. The thing we want to remember that's also happening here, right? The other thing that sodium was doing that was important was that it was being pumped out into the medulla, right? In that ascending limb, the thick part of the ascending limb. And then we're going through our distal tubule and into the collecting duct, which is dipping back down into the medulla. Okay. So about 70% of the sodium that we filtered in, I guess I can draw some arrows here, right? We're reabsorbing, we're reabsorbing, we're reabsorbing. So it's coming out of the fluid. So about 70% of the sodium that went into the nephron originally came from the plasma, freely filtered into the nephron, is getting reabsorbed by the body. Most of it is coming from the tubules and the collecting duct. I just drew on the loop of Henle because it's important we remember that it's part of that function of the medulla, although that doesn't substantially add to this 70%. Okay. Sodium is not secreted. So we remember secretion, right, is moving into the tubule, right? So late in the game, moving into the tubule, often in the collecting duct, maybe in the distal tubule, all right? That's not happening with sodium, but we will see that it does happen with potassium, which is why this is specified here. Okay. Now, the level of reabsorption that we're going to see is going to be regulated by hormones. It's going to be regulated by the two hormones we're talking about today, aldosterone and AMP, and we will be talking about them in that order. The regulation is happening in specific cells, actually the same specific cells where we were regulating our water reabsorption, which are called our principal cells. And they're in the same places as they were before, in the distal tubules and in the collecting ducts. When we're in the distal tubules and in the collecting ducts, this is going to be active reabsorption. So it's going to be responding to these hormones actively, and we'll take a look at exactly what's happening. On the whole, we're going to expect to see our sodium being reabsorbed through function of the sodium-potassium pump, which we've seen many times before. As we go through this, I want you to appreciate, right, that sodium balance and potassium balance are kind of going to be in the inverse of each other, because we're using this sodium potassium pump, which means that if we're moving sodium in one direction, right, that means we're going to be moving potassium in the opposite direction, just because that's how this transporter works, right? That's what this protein does. So when we move on to our discussion of potassium, that's an important thing to realize, and I'll highlight it then, but helpful to know at the outset so you can kind of piece that together as we go through. So here's a picture of what's happening in the proximal tubule. So in the proximal tubule, just because we haven't talked about the cell types in a while. So in the proximal tubule, we're looking at an epithelial cell. So that's a slightly different type of cell than the principal cell. And the epithelial cells are what we're focused on in the proximal tubule. The principal cells are more what we're focused on in the distal tubules and in the collecting duct. So sodium is getting reabsorbed here. We're using our sodium-potassium pump, right? We're pumping sodium into the peritubular fluid, ending up with high sodium here in our peritubular fluid, so the space between our epithelial cell and our peritubular capillaries, which allows sodium to move down a gradient from high to low. So then we have passive reabsorption happening here into the plasma. Okay. Sodium reabsorption here is important in part because it's part of what is allowing us to move other things through our proximal tubule epithelial cells. So glucose was the thing we really focused on in the proximal tubule, so that's what X would be here. So it's important that we keep low sodium inside our epithelial cells in the proximal tubule, because that's what allows us to do secondary active transport of things like glucose and this anti-transport, basically, right? This opposite movement, secreting hydrogen ions into the tubular fluid, right? So low sodium in the epithelial cell allows us to power movement across the apical membrane here. So this is not where we see our influence of aldosterone, right? This is just kind of always happening, right? This is not really regulated by hormones in the proximal tubule. In the distal tubule is where we are going to see a bunch of the regulation. But to begin with, we want to focus on a few of the mechanisms, right? So we're going to see that we have, again, a sodium-potassium pump, increasing concentration of sodium in the peritubular fluid, moving sodium into the plasma. Chloride ions are following. And in the epithelial cells of the distal tubule, not the ones we're really going to be focusing on for the rest of this lecture, but they also do exist in the distal tubule. So we can see this fact that we're going to have opposite movement at the apical membrane of sodium and potassium. And that's because of the action of the sodium-potassium pump on the other side, right? The sodium-potassium pump was giving us lots of sodium here in the peritubular fluid, which means that we're having our potassium concentrated inside the cell. So potassium wants to move down its gradient, which is out into the tubular fluid. Sodium wants to move down its own gradient. Sodium was low in our epithelial cell, so it's relatively higher in the tubular fluid. So the gradient for sodium points in, so sodium moves into the cell. Okay, so this is the basics of what's going on before we start to think about the influence of our hormones. Okay. Ultimately, we are going to be focusing on the effects of aldosterone and how it influences how much sodium is getting reabsorbed. And also, conversely, we'll talk at the end of this lecture on how much potassium is being secreted, right? Because we're having opposite action of these two ions. Okay. We are going to see that there is kind of a long pathway that's going to get us to aldosterone. Aldosterone is going to be the end of something called our RAS system. So we're going to have a renin angiotensin kind of pathway, but we'll talk about that next. All of that is ultimately leading us here to an effect on the principal cells of our late distal tubule. Okay. So the effect of aldosterone is to increase reabsorption of sodium, right? So just like when we saw with ADH, we're going to see a kind of similar pathway. We have aldosterone swimming along in our plasma, right? And we'll talk about how it gets there and why it's there in a minute. But when aldosterone is in the plasma, it's going to get into the cell. It doesn't need a receptor because it is a steroid hormone. Or I should say it doesn't need a receptor on the external membrane, on that basolateral membrane. Because it's a steroid hormone, it's lipid soluble. Steroids are lipids. Aldosterone specifically happens to be nonpolar, so it can cross directly across this membrane without any help. So the first time we see an effect of aldosterone is going to be actually inside the principal cell in this cytosolic receptor, right? Cytosol just refers to the fact that it's in the cytoplasm, right? Okay. So the effect of the aldosterone affecting this receptor on the inside of the cell is going to happen in two places. So we got a 1A and a 1B. We're going to actually talk about the 1B first, because I think that's going to help us understand the flow a little better. So one of the two actions of aldosterone in these principal cells in the distal tubule and collecting duct is that it's increasing the number of sodium-potassium pumps on the basolateral membrane. All right, this means it's also going to be really increasing these gradients a bunch, right? So before we had just a couple of these, now we're going to really increase our sodium concentration here in the peritubular fluid. And we're going to seriously decrease our sodium concentration in the inside, in the cytosol of that principal cell. Okay. So at the same time as it's increasing these sodium-potassium pumps on the basolateral membrane, it's going to be opening or even creating more sodium transporters on the apical membrane. All right, so we're just going to focus on the sodium parts here. We will talk about the potassium parts. They're basically the inverse. So you can see what they're doing. I'm just going to trace through sodium for right now. Okay, so we have really lowered the sodium in the cell using this sodium potassium pump, which means that there's a really big gradient for sodium to flow into the cell, right? So we're going to have more sodium flowing into the cell, both because we have more transporters that allow for it, right? More of these open channels. And then we are going to have lots of pumping into this paratubular fluid, creating a really high concentration of sodium in the paratubular fluid, which ultimately means, right, we also have a big gradient across this membrane of the paratubular capillary. So between the paratubular fluid and the paratubular capillary, there's going to be a big difference in sodium. So sodium is also going to get reabsorbed into the plasma on this opposite side. So this is going to happen faster. It's going to happen more, right? We're increasing the sodium reabsorption. Okay. Ultimately, the aldosterone is coming from the adrenal cortex, but we're going to see that there's a really kind of complex flow of how we are releasing the aldosterone and why we are releasing the aldosterone, I should say. So we're going to get into those details in a second. But questions before we move on about what's going on with these membranes with these gradients? Okay. Okay, so here is the flow of how we are making this aldosterone, right? So it's the RAAS, or the renin-angiotensin aldosterone system. So really, if you can remember this word, right, this is telling us the flow. We're going to use renin to create angiotensin. We're actually going to create angiotensin 1 and then angiotensin 2. That's going to trigger a release of aldosterone from the adrenal cortex. And it's a system, that's the S, because we have so many steps here. Okay. So this is also going to use a lot of different organ systems, right? So we are going to be seeing a role for the liver. We're going to see a role for the kidney, right? Which makes sense because we're talking about the kidneys in this unit. Specifically, we're going to be looking at the juxtaglomerular cells, which we haven't seen in a while, but those are these ones, right? Kind of stuck here by our glomerulus between those arterial cells, and touching the tubule and the arterioles pretty much. Okay. We're also going to be affecting the adrenal glands, which, as a quick reminder, right, sit right on top of your kidney, which is helpful because we ultimately want to see that aldosterone kind of going into the kidney and affecting those principal cells in the tubules on the inside of the kidney, right? So that close relationship is actually pretty helpful for us. Okay. So the granular cells of the juxtaglomerular apparatus are going to secrete renin. So we're remembering that we have these different types of cells here kind of on our arterioles, right? So we have some glandular cells here on our afferent arteriole, which was the one bringing blood to the glomerulus, right? We have some more granular cells all kind of in here, right? We also had our macula densa cells embedded in our distal tubule here in this juxtaglomerular this juxtaglomerular apparatus, or this juxtaglomerular complex. Those are equivalent terms, right? Remember, there were also mesangial cells here. Mesangial cells were one of the ways we were regulating flow into the glomerulus, right? It was one of our regulatory mechanisms for the GFR. Okay. But it's the granular cells really, that are secreting renin. So overall, kind of what's happening actually is that the macula densa cells are sensing changes in sodium. The granular cells are going to react by secreting renin. So we're jumping in here to this pathway. And what renin does is renin is going to convert angiotensin to angiotensin 1. So this should kind of remind you of when we talked about clotting, right? We had a lot of different clotting factors flowing along in our bloodstream all the time. That's kind of what's going on here with our RAS, right? Our renin, angiotensin, aldosterone system. So all the time, right? Our liver is making angiotensin and it's flowing around, right? So it's just flowing around, right? Other things that we have happening all the time is that in the walls of the capillary endothelial cells, we have these ACE cells, or these ACE proteins, I should say. So ACE stands for angiotensin converting enzyme. So they're kind of stuck in the endothelial cells, but they're going to need the help of angiotensin 1 to convert the angiotensin 1 to angiotensin 2. Okay. So renin got secreted. It converted the angiotensin inogen from the liver that's circulating all the time into angiotensin 1. Okay. Then we are converting to angiotensin 2 using these ACE, right, the angiotensin converting enzymes that are in the endothelial cells. And they're not doing this until we have renin, because until we have renin, we don't have angiotensin 1. Okay, so they are there all the time. Okay, then once we have angiotensin 1 converted to angiotensin 2, that gets us into the adrenal cortex, and that angiotensin 2 is what tells the adrenal cortex to release aldosterone. Are these numbered well? Okay. Yeah, so I would say 1 would be the secretion of renin. I might then jump down here and say our second step is the conversion of angiotensinogen into angiotensin 1 by renin. And then 3 would be converting angiotensin 1 to angiotensin 2. 4 would be converting angiotensin 2 into aldosterone. So that's a useful flow, and these middle points are kind of telling you other things you need to know to understand where that angiotensinogen is coming from and what ACE is. Okay. A note here, you may have heard of ACE before. Some people take something called ACE inhibitors, right? So ACE inhibitors target this system by inhibiting or blocking, stopping, right? This angiotensin converting enzyme, right? And if we block that, then we're ultimately going to be blocking the release of aldosterone. So sometimes we're not going to want to release aldosterone, and these medications, these ACE inhibitors, are usually prescribed in response to consistently high blood pressure, right? Pathologically high blood pressure or heart failure, which is related to blood pressure, because of the effects that we're going to see of aldosterone on sodium reabsorption and then ultimately on blood pressure. So there are big pathways, and really whenever we have medications that we take, they're affecting some part of one of these physiological systems that we've been talking about in class. This is just a common one. Okay. So here's a zoomed in picture of what's going on. If I were to number the steps directly related to renin, right, we'd pick renin released by our juxtaglomerular cells, allowing us to get angiotensin 1, which came from this precursor angiotensinogen. Then we're converting to angiotensin 2, which is affecting the adrenal cortex, giving us aldosterone. Okay. That aldosterone is how we then got to what we were looking at in the principal cells. I'm going to flip back for us just while I talk, right? So that's the aldosterone that we saw here that's having our kind of 5th and 6th effects here, right, of increasing these basolateral membrane pumps and opening these sodium channels in the apical membrane. So ultimately, if we look at this, the release of renin affects the reabsorption of sodium. So if we have a bunch of renin, we're going to be reabsorbing more sodium. Here we have a diagram that's focusing on a specific point, which is angiotensin 2. So that's the step right before our aldosterone. And angiotensin 2, we've actually seen this name before. There's a couple of different effects. So what we have seen before, though we didn't focus on it much, was that one of the effects of angiotensin 2 is directly causing vasoconstriction in systemic arterioles. So angiotensin 2 uses this system to increase MAP, so to increase blood pressure. So you can immediately see why, if you have high blood pressure, you might want to turn off this system, right? Okay. Now, the more complex kind of pathway that we're going to be thinking about with this system is, note how we have this kind of loop going on here. So ultimately, this system is supposed to be reacting to really drops in blood pressure, drops in blood flow. But it takes a lot of steps to get there, and that's not immediately obvious from its effects on sodium. So I'm going to kind of trace us through there. So our angiotensin 2 is creating our aldosterone secretion, right, in the adrenal cortex. We saw that on the previous slide. And then we go to our principal cells. We're increasing sodium reabsorption. That much is hopefully, at least after you study this flow a little bit, kind of clear. Now, what's not immediately obvious, right, is how this effect of increasing sodium reabsorption has anything to do with blood pressure, right? And to do that, that's when we need to link up our understanding of aldosterone with our understanding of ADH. So as we increase sodium reabsorption, right, what we're doing here when we increase sodium reabsorption is we're increasing the sodium concentration in the plasma, right? So we're making our blood saltier. Now, that is not our ultimate goal, actually, right? The reason we want to increase this sodium in the plasma is that we're trying to trigger the reabsorption of water. And so what we're doing here really is that we're increasing sodium plasma so that we increase the extracellular fluid osmolarity, which we remember is one of the things that are sensed by those osmoreceptors and the hypothalamus, right? So when your brain, right, senses that we have a lot of sodium in the blood, it's going to release ADH, right? It doesn't want salty blood. So by activating this system, by activating aldosterone, ultimately, we're activating our posterior pituitary to secrete ADH, which increases water reabsorption in the distal tubules and collecting ducts because it's inserting those aquaporin 2 molecules in the apical membrane, which ultimately means we're increasing plasma volume. If you have a lot of plasma volume, if you have a lot of blood in your circulation, that means you are raising blood pressure, you're raising MAP. Okay. So this is kind of complex, right? So we're using this one system, starting with renin. So we go renin, angiotensin 1, angiotensin 2, aldosterone. Aldosterone basically creates sodium in the plasma. Sodium then triggers ADH secretion, which triggers water reabsorption. So it's a really long pathway, but it's a really important one because we target this with basically a lot of different kind of medications and processes, right? So we have ACE inhibitors that affect the actual creation, right, of angiotensin 2, but we also have diuretics, right, which target ADH secretion or water reabsorption, right? So we can target kind of this part of the flow. Ultimately, our goal is to affect the cardiovascular system, affect blood volume, blood pressure, which can help ease stress on our blood vessels, but importantly on our heart. So final cool effect of angiotensin 2 is that another thing that's happening in response to them is that we directly stimulate thirst. So when you get thirsty, one of the things that may be happening, right, is renin secretion converting to angiotensin 1, converting to angiotensin 2, stimulating you to want to drink. Because then when you drink water, also increases the amount of plasma you have, right? That water gets into your plasma, ultimately raising your blood pressure, which I think is pretty neat because I don't always think of observable behaviors as being controlled in such visible ways by hormones. I think that's nifty. Okay. So some stimuli for this release of renin are the potentially decreased pressure in the afferent arteriole. So remember, we're targeting the release of renin at this juxtaglomerular complex, right? So if it senses that you don't have enough blood coming into the kidney, that would be a signifier of low blood pressure, low blood volume, right? If you don't have enough blood flow here. So ultimately, we know this pathway is going to raise our blood volume, raise our blood pressure. So that's a good signal that we should be releasing renin. Another thing, an extrinsic factor that affects the release of renin is sympathetic nerves to the kidney. So renal sympathetic nerves also tell you to release renin. And also decreases in sodium and chloride ions in the distal tubule filtrate. Okay. So here, if we have decreases in sodium leaving, we can sense that because we're stuck here between our arterioles and the distal tubule. This part is not immediately obvious why this happens. So I kind of want to explain that out for you right before we go. I know we're nearly at time. But what's kind of happening here, right, is if you have low sodium, right, so a reason you would have low sodium in the filtrate, right, is that filtrate, all this fluid is moving through your nephron, right? And we have certain amounts of sodium that we have to reabsorb, right? So we have to reabsorb sodium, for example, in the loop of Henle, because we're pumping it into the medulla to keep it salty. So if we note that there's not very much sodium in the fluid that's passing by here, passing this juxtaglomerular complex, that can be an indicator that we don't have kind of fast enough flow or enough flow through the nephron as a whole. And so we are reabsorbing the amount of sodium and chloride ions that we have to, but that's dropping down the concentration of those things in our urine too much because there's overall not enough fluid going through, which may be a result of the fact that we don't have enough blood flow coming into our kidneys, right? So that's how that's kind of linked up to our blood pressure signal here. Okay, so I will let you go for now. I do want to make sure we get through the end of this packet and also have time to review. So we will finish this up next time we meet. I will try maybe to make a video for you guys over the weekend on this stuff, which will be optional to watch, but I know you may want a little bit of a jump on your studying. Okay. Have a great weekend, everyone. Yes, I will put a quiz up shortly.